Next organelle, the genome. Genome, the entire set of genetic material present in a cell. So in case with bacterial cell, as we know, they are prokaryotic in nature. Prokaryotic means, or prokaryon means a cell in which the nuclear membrane which is not well developed. So it is having, or it is lacking, the true nucleus. The DNA in that case is freely suspended inside a cytosol. In case with bacterial cell, the genome or genetic material is present in single chromosome. While in case with the evolved organisms or in eukaryotic organism, this chromosome is divided into different chromosomes, or the genome is divided into several chromosomes. So for example, in case with human beings, we have 23 pairs of chromosomes which is present in nucleus. In case with prokaryotic cell, the genome is a single chromosome which is double stranded circular genome. What does this chromosome has, or what does this DNA has? It has the genes present in it. So what is the genes? They are the segment of the genome which codes for functional product. That is, when allowed to express, this information from DNA can be converted into mRNA by the process of transcription. If allowed, it can be converted from mRNA to protein by the process of translation. So this process is regarded as transcription and translation. We can call it as an protein synthesis. So this DNA, it consists of sequence of nucleotides. So we know that in case with DNA there are 4 nucleotides, A, T, G and C. But the sequence of this nucleotide, it carries information. So that information is in the form of genes. So genes are the segment of the chromosome or DNA which codes for functional product. That is, it has information in the form of code, or we can call it as an codon. You might have studied the genetic code. The information which is present in the form of codon present on mRNA is decoded by tRNA, and the information, that is the translation of that information, is done into the amino acids. So for that, rRNA it helps, that is ribosome it helps, tRNA it helps to convert, to translate the nucleotide language into amino acid language. Therefore we call it as a translation, or we call it as a protein synthesis. So inside a bacterial cell, the DNA is freely suspended inside a cytosol which is having single chromosome, double stranded circular genome is there. It carries genes in it. These genes are nothing but segment of DNA. When allowed to express, it can convert itself into mRNA and further translate into protein. Now they are free in cytosol, that is what we have discussed. They can be attached to the plasma membrane. In case with E. coli, what is E. coli? E. coli is Escherichia coli. It is one of the model organism that is used for the study in bacteriology. So this Escherichia coli, Escherichia is the genus and coli is the species. So we can write it, this type of nomenclature is regarded as an binomial nomenclature system. So we can write it as Escherichia coli, or we can simply write it as E. coli. So E. coli, a model organism, it has around 4 × 10⁶ base pair genome, and out of this genome, that is, this many number of nitrogen base pairs are present in that organism. 4 × 10⁶. Base pair means, we know that Chargaff rule, A adenine it pairs with the thymine and guanine it pairs with the cytosine. So this base pairing, altogether in an E. coli there is 4 into 10 raised to 6 base pair. So that much length of DNA is present in E. coli. Out of that 4 into 10 raised to 6 base pair, there are around 4000 genes, that means which can express into mRNA and protein, which codes for functional product. So in case with a typical bacterial cell E. coli, it has 4000 genes. So genes, they are present on DNA, that is they are present on chromosome. Genes can be expressed if proteins are needed. Now this can be regulated, that is the genes, they can be converted, they can be translated into protein by the process of transcription followed by translation. But depending upon the need of a cell or surrounding, the copy of that proteins can be vary, that is at a particular time if the product of the gene is not needed, the genes can be repressed, or it can be not allowed from expressing. If needed, it can be allowed to express, that is what we call it as a regulation. So genes are under regulation. For example, lacoperon, you might have studied, it is an utilization of lactose sugar. For that 3 genes are required. So if lactose is present in the media, only then that operon will function, otherwise that operon is in a repressed state. So genes, they carries information, but they can be regulated depending upon the cellular need. Next thing is plasmid. Plasmid, they are extrachromosomal circular DNA. Extrachromosomal. So chromosome means the genome, the genetic material, that is the identity of that particular cell. In case with bacterial cell, apart from that genome, it has some circular component, this circular double-stranded DNA, which is apart from this bacterial genome, it is present inside a cell. Most of the bacteria, they have plasmid in it. So what that plasmid is, they are circular double-stranded circular extrachromosomal DNA. If you compare with the size, they are comparatively very small in size, much smaller than that of bacterial chromosome. It carries extra genes in it, as we are calling it as a double-stranded DNA. We know that DNA is made up of nucleotide, and if nucleotide is there, again the sequence of nucleotide will carry some information, that is it will carry genes. That plasmid also carries genes in it. Some of the genes may be beneficial for that particular cell. Like for example, some bacteria has enzymes or some genes which are present on the plasmid, which helps it to survive to a particular condition. For example, antibiotic resistance gene. If say for example ampicillin resistant gene, ampicillin is one of the antibiotic. If a bacterial cell, it has a plasmid, and on that plasmid it carries ampicillin resistant gene, that is a gene which when allowed to express, it can synthesize the product which will be able to hydrolyze the ampicillin or break down the ampicillin. So if this plasmid carrying ampicillin resistant gene is present in this bacterial cell, this bacteria will carry additional favor, that it can grow even in ampicillin containing media because of the presence of ampicillin resistant gene. Likewise, Ti-plasmid we know, tumor inducing in case with the Acrobacterium tumefacium. So that plasmid, if it is having that gene, it will help in nitrogen fixation. So plasmid, they are circular extrachromosomal DNA present in a bacterial cell. They are comparatively very smaller to the size of bacterial chromosome. holds extra beneficial genes for that cell. One or several kinds of plasmids can be present inside a cell. The plasmid, we know about the different types of plasmid, like F-plasmid. F-plasmid is helpful in fertility, in the process of conjugation. That is F-plasmid. With the help of sex pili, the chromosome or the plasmid can be transferred from one cell to other cell. So F plus, if the bacteria carries that F-plasmid, we call it as an F plus bacteria. If the bacteria is devoid of that plasmid, we call it as an F minus bacteria. The bacteria is also known as donor, and the other one which is lacking it is known as recipient. And with the help of sex pili, that plasmid can move across the bacterial cell. Likewise, there is another plasmid which carries Col gene, that is colchicine resistant genes. If that gene is present on that bacteria and on that plasmid, that bacteria will be on a favorable side. Likewise, there is R-plasmid that we have discussed, a bacteria carrying antibiotic resistance gene in it. So there can be several different types of plasmid, and the copy number also, it can varies depending upon the cell. In some bacterial cell, the plasmid copy can be restricted to 2 to 5 in number, while in some cells the copies can be up to 100. So this extrachromosomal DNA, it can be present in limited number, a low copy number, or it can be high copy number. A few or many copies of such kinds of bacteria can be present, and they are regularly used into the process of genetic engineering. That is, in recombinant DNA technology, this plasmid, they act as a vehicle, it acts as a vector. So the gene of our interest can be added or loaded on the surface of plasmid, and this plasmid, if allowed to conjugate, or if we allow that bacteria, or we transfer this plasmid to a bacterial cell, that the bacteria will gain the additional information or that additional characteristics. So in case with rDNA technology, that is recombinant DNA technology, genetic engineering, we use this plasmid.